Okay, so in this lecture series, we are going to talk about different neurotransmitters and receptors. In the slides for class, I think there are about 10 slides of review to just talk about the different receptor types, ionotropic versus metabotropic. And if you guys remember, the ionotropic receptors are ones that we have fewer of, and they work directly on ions, whereas the metabotropic, we have about 80%. They're slower. 80% of our receptor types are metabotropic. They're slower in terms of getting an effect. Also talked about IPSPs and EPSPs, excitatory versus inhibitory postsynaptic potentials, and these sort of add up in terms of stimulation to determine, is that neuron going to fire the action potential. So I do encourage you to go back and look at some of that. There's another brief video that I did on it, but if you would like to look at the chapter material, I highly suggest that you just read up on that just as a reminder before you get into these neurotransmitters and chemicals, because it will help you if you've forgotten what some of these terms mean. So today what we're going to do is I'm going to split this video probably into two. We're going to talk about monoamines and amino acids. I think I'll probably stop at the amino acids maybe, just so that we can kind of not go into a really long video. So with the monoamines, we're going to talk about dopamine, norepinephrine, serotonin, and histamine. With the amino acids, we're going to talk about glutamate, GABA, and glycine. We're also going to talk about some peptides, some lipids, nucleosides, and finally end with some soluble gases. So just to start with the monoamines, this is a question that I've gotten in class before. How is it that we talk about the receptor being the lock and the neurotransmitter being the key? And how is it this works? And how is it that some agonists or antagonists mimic the shape? There's a lot of questions about what is the shape of a neurotransmitter. Well, this is why I've got these sort of molecular structures here to show you. So like this is the shape of dopamine. That's the sort of what you look when we look at sort of molecularly, these are the kinds of atoms. The kind of shape looks like this. This is how the chemical is going to fit into the receptor type because of the shape of these atoms. And so like this is a dopamine. We have norepinephrine here and epinephrine here, as you can see chemically how these sort of atoms make up. And we look at other drugs that are kind of mimicking these, they're going to try to look at the very similar shape to be able to fit into the receptor types. And also we have sort of sub-receptor types that are able to be a little bit more flexible and take different kinds of neurotransmitters or chemicals in. Here we have acetylcholine. As another example, we have serotonin here. And as you can see, even the monoamines are broken up into different kind of amines, just so that you guys can see that we can categorize these chemicals based off of structure, based off of molecular structure or type, as one of the ways that we're going to classify these. So just looking at the amino acid neurotransmitters, we have glutamate, aspartate, GABA, and glycine, and we're going to go into a lot of these sort of individually so you guys can kind of see. I'm just showing you the structure. I would never ever expect you to know what this breakdown is. This is just to show you what it looks like, because I got so many questions about, well, what makes up the molecular shape? And because we're not going into biochem stuff, because we're a general basic biopsych class, I do not want you guys to have to memorize all this. What I just want you to see is how the shapes differ based from, for example, glutamate to aspartate to GABA. You can see how, you know, the molecular structure is different, meaning that they're going to look differently. Here is an example of a peptide neurotransmitter. We call these neuropeptides. These are made up of short amino acid chains. And so here's an example of tyrosine, glycine, for example, the different makeups of these amino acids, the parts. Okay, so that's not glycine, it's just the parts and how they're making up the different neuropeptides. So again, I don't expect that you would know, be able to draw this out, for example. I never would expect that. I just want to show you that to see how it looks for just sort of basic purposes. So let's talk about acetylcholine, because it's kind of its own little class. And it is one that's very, very important for the brain because it's involved in our ability to use our muscles. Okay, and so acetylcholine is that main transmitter that's released by our motor neurons, and there are several poisons that interfere with acetylcholine signaling that can cause you to spasm with too much acetylcholine, for example, black widow venom. Or it could interfere with the signaling and you don't have enough, and that could paralyze you, like the botulinum toxin, and that's where we get botulism poisoning, and that can actually make you, that can paralyze you and can kill you because of the motor signaling that's important for things like breathing can stop, and obviously that can make you stop breathing and make you die. With acetylcholine, it's also really important for attention and maintaining attention and alertness. It's also really important for learning and memory. And it's part of the process of the cells being able to secure more connections and network better, and to be able to, for example, learn a new task as well as learn new information. So it's really important when it comes to memory. Acetylcholine is one of those main neurotransmitters that we know quite a bit about. We know about 50 to 60 neurotransmitters we know that exist in the brain, and we know they have something to do with something, but we're always finding new information. Even now, you would think that we would know everything about the brain. There's still a lot that we don't know. So this is an example. So all these brains that you're going to see that are kind of shaped like oddly like this, these are rat brains. And so you can see the way that it's like positioned. If you were to expect this into a human brain, you would expect it to be more upright because the cerebellum is kind of on the back of the brain. Whereas with a rat brain, because the head looks like this, it's a terrible drawing, but you see what I'm saying. That is something that you can see that the shape of the brain should look different. That should be something that you're like, wow, that looks weird. That's because it's not a human brain. So the interesting thing about rodent brains, though, is that they're very similar. They have a lot of the same structures. They're slightly different in terms of location, obviously, because this one is the way that it's sort of shown. But a lot of the same structures there. So all these pictures that are going to show the pathways are of rat brains. So this is a schematic of the acetylcholinergic neurons and how they distribute throughout the brain. So as you can see, there are a lot of pathways that contain receptors for acetylcholine. So if you were to get, for example, a toxin that's going to impact this neurotransmitter, it's going to impact the brain completely at a whole level. It's going to go everywhere because we have so many different kinds of pathways that have this particular neurotransmitter being, sort of the primary one. So it's really important that we're careful about acetylcholine and making sure that it's, of course, balanced. So this is an example of the different types of receptors. And so an acetylcholinergic receptor, there's two main types of receptors that have places for acetylcholine. One is a nicotinic receptor, which is what we see here, for example. And you can see this is the binding site. This is an example of an ionotropic neurotransmitter. So, it is a very fast, we only have about 20% of these of all of our receptor types. It's quick binding for the entering of ions into the receptor. It's, as you can tell, it's a very specific shape of a receptor type. And of course, these are the binding sites specifically for acetylcholine. Now, we also have muscarinic receptors, which are metabotropic. They're slower, but they do make up more of the receptor types. So I don't have a separate picture for that, but know that it is a different, it's a bit slower in terms of receptor for acetylcholine. A couple of other monoamine neurotransmitters. So we have acetylcholine, a group, which is dopamine, epinephrine, and norepinephrine. And so here you can see the way that these catecholamines are synthesized. And so, again, I don't expect you to memorize everything by heart. What I want you to do is know about the synthesis of different chemicals in the brain, because and how does that relate to what we know about the brain, and why is it important? Well, we've already talked about, for example, dopamine. So dopamine can't be administered, right? We can't give somebody dopamine and then expect them to increase their dopamine. Dopamine does not cross the blood-brain barrier. So when we talk about synthesizing dopamine, what do we have to start with? Well, tyrosine is one of the precursors of dopamine. And so tyrosine can be sort of manufactured into dopa to be able to give us the precursor to dopamine, so that if we were to administer a synthetic version, the person would essentially be making more dopamine, synthesizing more dopamine, because drugs can do different things to different parts of the brain, and we talked about this. So, if the drug is going to serve as an agonist, it can serve as an agonist on any level of the synapse. It can make you release more, for example, dopamine. It can make you it can block reuptake so that you have more dopamine in the synaptic cleft. It can cause you to create more dopamine, to synthesize more dopamine. So it starts at the very beginning and says, we're going to make more of this. It's like, baking a cake. Here are all the ingredients. Here, I've provided everything for you. Now you could go ahead and make your own cake. That's kind of the idea behind L-Dopa, which we'll talk about in a minute. It is the precursor to dopamine, which makes your brain make more dopamine, which means that you have more dopamine in the brain. So this whole process of synthesis, though, it's got to start with something, and it starts with your diet, what you eat, the food that you take in. For example, might have tyrosine, and then you eat that and you consume it, and your body processes it and then synthesizes it into chemicals and neurotransmitters for your brain to use. So understanding the synthesis process, I'm not like, oh, okay, well, memorize that you start with tyrosine and then go to dopa and dopamine and then norepinephrine and epinephrine. I'm less concerned with you memorizing every single thing, because you get to use your notes for the test. So it's not like you guys have to memorize this by heart. What I do want you to do is know that there are precursors in the synthesis process for every chemical. And that is important because that's what we know about how our brains use these chemicals. So, I hope that, it eases some of the, probably anxiety of seeing a lot of these chemical structures and seeing the synthesis process. I want you to know it at a more sort of critical thinking level. And if you look at the endolamines like serotonin and histamine, a lot of people are familiar with these kinds of chemicals. But again, there's a synthesis process to all of these chemicals. So let's look at the dopamine system. This is one that we're going to spend a little bit more time on, because a lot of people are very curious about dopamine. We know a lot about it. There are several dopamine systems throughout the brain. So we talked about this idea of, if you disrupt a system, if you take a person and you give them the ability to make more dopamine, or the ability to process more dopamine, what is that doing? Well, if you remember correctly, I talked about how it impacts other chemical systems, but also lots of chemical pathways have multiple pathways in the brain through different systems. So like, for example, there's a mesocortical pathway system of dopamine. Okay, this is one of the dopamine systems. This isn't the dopamine system, it's one of them. Okay, and it moves through the mesencephalon and into the midbrain. For example, the ventral tegmental area. So here it's sort of start process here, and then moves its way throughout the brain all the way to the prefrontal cortex, and the neocortex. And so we know that the prefrontal cortex is really important for decision making, problem solving, and planning. The mesocortical pathway along that has important structures that are important for forming short-term memories. Memory in general. It's, here we know that the hippocampus is really important for memory. It's important for planning. We also know that when you have too much dopamine in the system, into this particular mesocortical system, we see issues with a person developing symptoms for schizophrenia. And so if you look at, some of these structures, planning, problem solving, memory. We see individuals with schizophrenia having issues with all of these things. They also may experience things like hallucinations and sensation and perception abnormalities. And all of this is kind of involved in some of these dopamine pathways, specifically in the mesocortical area. So if a person is having too much dopamine, it can impact the system. And I'm going to give you an example. It's jumping ahead just a little bit. We're going to get to this in just a second, but I really want to demonstrate that, if you have a system that is not getting enough dopamine, okay, like for example, with Parkinson's disease, which we're about to talk about in a minute, and you flood the system with dopamine, specifically the mesocortical system, because remember, you can't pick a system. If you're having the person take more dopamine, like L-DOPA, to synthesize more dopamine, you're going to affect all the systems involved with dopamine. So you're going to affect the mesocortical system. Well, if you have a person with Parkinson's and you overdose them with too much L-DOPA, now you're going to start to see symptoms of schizophrenia, because again, this is overflooding these pathways with dopamine. So like the thalamus, for example, we see these projections to the thalamus. The thalamus is really important for filtering out those additional sensations, those additional somatosensory feelings. And so, this is where we might somebody might start experiencing some hallucinations, or, visual hallucinations, or physical sensations that aren't there. So we do have to be careful with this, system. It's got to be very balanced. Another of the dopamine system is through the mesolimbic area. So the mesolimbic area is important for reward and reinforcement. If you guys remember from the previous lecture, we talk about the ventral tegmental area being really important for the continued use of drugs, for example, addictive drugs. It's this particular area, looking at the mesencephalon, the midbrain, containing that ventral tegmental area, is important for, this addiction pathway. So we talked about the nucleus accumbens, right? We talk about that with initial use, and then we talk about the ventral tegmental area being important for continued use. So this is like where your habitual learning takes place. This is where habit forming and procedural learning take place. And so this is where you're starting to see people become addicted to the process of taking drugs, the people associated with taking drugs. We see incentive salience for the drug paraphernalia used to administer drugs, right? All of this is the same sort of pathway that we see all of this happening. So it's important for reward. It's important for reinforcement. We know that reinforcement is really important for learning. So this particular pathway is really implicated in addiction. And it also, if you look at, kind of addictive drugs, for example, hijacking the system, we see that it also hijacks the limbic system. The limbic system has the amygdala, the nucleus accumbens, we talked about initial drug use there, as well as the hippocampus. So we see it also impacting memory. Memory is involved in learning, and all of these sort of systems that work together. Now, another thing that I want to talk about here, because we know that we jumped ahead and we talked about Parkinson's, but this is really important to talk about all of this sort of together, okay? So we talked about Parkinson's disease being a disease where the neurons that make dopamine essentially atrophy or become damaged and die, okay? So we see the nigrostriatal area being really important. So this is the next pathway that we're going to talk about, which is through the substantia nigra. And I talked about in previous lectures that the substantia nigra is dark in color. And the reason why, if you were to do an autopsy on a person and they had a healthy, intact substantia nigra, this particular area of the brain is dark in color because it's producing dopamine in these areas. And dopamine is it just so happens that these particular areas are darker in color. And then one of the first autopsies produced in conducted on a person who had symptoms of Parkinson's disease, the doctor actually discovered that the substantia nigra was not dark in color, and this person had these symptoms of problems initiating movement, which is what Parkinson's disease is. It's a motor condition where these substantia nigra neurons are essentially not able to produce dopamine, and they're not able to send the right signals to initiate movement. So people with Parkinson's disease may be confined to a wheelchair. They have a lot of difficulty with motivating that initial movement, and once they get moving, they do kind of like, for example, a shuffle when they walk. There's a lot of other things that go along in the brain there, but that's, kind of the primary thing. And this is going through that nigrostriatal area, this particular pathway. So you can see here going through the global pallidus and the caudate nucleus, putamen, things like that. And this is the particular striatum, that pathway that follows, that contains, like I said, the caudate nucleus and putamen, which is what's right here. Now, what's really interesting is we talked about the fact that, dopamine is something that people with Parkinson's need. So what do? You give them a drug called L-Dopa, which is the precursor to dopamine, and have them make more dopamine. And we talked about this system here, okay, the mesolimbic system being really important for reward and punishment. And we talked about the system before that being important for remember, these are all three dopamine systems, mesocortical area being important for memories, planning, problem solving, associated with schizophrenia, because it has these projections, to the prefrontal cortex. Okay, so let's take all of this together. You have a person who has Parkinson's disease, and you give them more dopamine. So it's going to, or give them the precursor to dopamine, which means they're going to make more dopamine. So now what happens is that dopamine is going to affect the mesocortical, the mesolimbic, and the nigrostriatal area. It's going to impact each of these three systems. You can't just single out and say, well, we're only going to give dopamine in this pathway. It's going to impact all of these pathways and all of these connections and all these projections. So a question that I got in class the other day, which it's funny because I look back and I think about, sometimes I get like almost blindsided, even though it was a great question and I should have known better. As I was looking through some of the literature, it hit me. I already knew the answer to this and I completely blanked on it. So a student asked me in class, well, okay, you say that a person with Parkinson's disease is lacking dopamine. What does that have to do with what we know about addiction? And so what's really interesting is that there's a lot of literature talking about how if a person has Parkinson's disease and you up their dopamine by giving them L-Dopa, they're creating more dopamine. Some cases, and this isn't rare, this does happen to some people, they'll actually develop habit, like addictions. So they'll might develop a gambling addiction. They might become addicted to certain drugs. They might develop a sex addiction or an eating addiction. And it's because we're seeing these systems being activated with more dopamine. It doesn't happen to everybody with Parkinson's disease, but it is an interesting side effect of some of the medications of once they're being treated for this particular disease. So yes, we can see changes in addictive behavior in people with Parkinson's disease once they're actually treated for their condition. So it was a really interesting question that I got that I completely blanked on. Okay, so let's finish up dopamine, just talking about the different receptor types. So there's whole families of receptors. So we have D1 through D5. D1 family is all the receptors are on the postsynaptic side of the cell. Remember we talked about like having... giving out more dopamine, the next cell taking it. That's like the postsynaptic side. And then the D2 family contains both pre and postsynaptic receptor types. So we're talking about the cells being able to reuptake dopamine as well as send it to the next cell. And all of these receptor types are metabotropic. So they're, the kind of slower types. So let's talk about norepinephrine and epinephrine. So norepinephrine is also known as noradrenaline. It's the same exact chemical sequence. It's just a different word for it. And when we talk about epinephrine, epinephrine is also known as adrenaline. Again, epinephrine and adrenaline are the same thing. Norepinephrine and noradrenaline are the same thing. So norepinephrine is much more important in the brain than epinephrine. We know that norepinephrine is found extensively in the autonomic nervous system, which is a part of that peripheral nervous system. Also does have a really important role in arousal and attention in the brain. It's really important for keeping us alert and awake. And what we see here is, when we're looking at a terminal button, for example, remember that the vesicles, these are these like pockets of chemicals. The way that norepinephrine is synthesized is actually synthesized inside the vesicles themselves. It's actually made in the vesicles from dopamine specifically. And so, that's just, we talked about different ways that a chemical could be synthesized and then could be transported through the microtubules that are located in the axon. But these norepinephrine is specifically made inside the vesicles directly from dopamine. And then epinephrine is also found in the autonomic nervous system, but it's not as important in terms of the brain function as norepinephrine. So a lot of people realize, okay, adrenaline hypes you up and gets you all excited because they're more familiar with that in terms of the autonomic nervous system. Just some of those noradrenergic projections. So these are cells that have the ability to have norepinephrine or noradrenaline receptor types. So we see that there are projections all throughout the brain. Again, this is a rat brain. We see that the pons and the medulla have cell bodies that are specifically have the receptors for noradrenaline, and the locus coeruleus here, and they can project throughout the brain. So remember, noradrenaline is much more important than adrenaline, or norepinephrine is much more important. Receptors for epinephrine and norepinephrine are called adrenergic, adrenergic for those particular receptor types. They're located both in the brain and the peripheral nervous system, because remember, the peripheral nervous system is important for the autonomic nervous system. There are 4 major types, 2 alphas and 2 betas. All of these are metabotropic, but the presynaptic, so there's presynaptic and postsynaptic. The presynaptic autoreceptors are located at the alpha 2 receptor types. So let's talk about serotonin. So you might see serotonin abbreviated as 5-hydroxytryptamine, but whenever you see 5-HT, that's serotonin. Serotonin has a lot of really important functions in the brain, a lot of roles in the brain. It's synthesized from tryptophan. If you guys remember, we had talked about tryptophan being in turkey. And so there's this whole thing of, whenever I eat a giant turkey dinner, I get really tired, for example, during Thanksgiving. And it must be the tryptophan because I'm synthesizing it to serotonin, and serotonin is really important for mood and for sleep and for appetite. And so, I'm getting hungry, and then I'm nice and satisfied and I'm in a good mood and I feel sleepy. If that helps you remember that tryptophan is synthesized into serotonin, then great. Serotonin, it is then synthesized into serotonin. If that's what helps you, that's awesome. Just go ahead and remember it that way. But realize that a lot of times when we look at different kinds of like amounts of tryptophan in turkey, it's probably not enough to really make you feel that sleepy, but it's usually the size of the dinner and the fact that you're not doing anything on vacation that makes you more sleepy. But like I said, if that helps you remember, then go for it. And you can kind of see how we get the names. So, the hydra, the oxy, the trypt. right? So amine, you can kind of see how we get the names based off of, the different structures. Tryptophan hydroxylase, right? Which goes into decarboxylase, which then gets, manufactured into specifically serotonin. You can kind of see how we're getting some of these root terms, like what I was talking about the beginning of the lecture. We also know that serotonin is really important for arousal and pain management. This is kind of like our happy, chemical that we try to think of as like that's what makes us feel good and happy. We have at least 9 different subtypes of receptors. All of them but one are metabotropic. So this 5-HT3 is ionotropic. So these, remember, are the slower receptor types, whereas ionotropic is much, much faster. These are the projections throughout the brain. It's synthesized specifically in the raphe nuclei. And if you know, if you take the sleep class with me, we talk a lot about serotonin being important for sleep, and this area right here is really important for consciousness and sleep. So serotonin is synthesized in the raphe nucleus, and then as you can see, there are quite a few extensive projections throughout the cortex. And the hippocampus is important for memory. The basal ganglia is important for movement. These are all different areas that are really important. Amygdala is important for regulation of like the fear response and emotion. Hypothalamus is really important for regulating your appetite. It's got a lot of nuclei there that's important for mood and sleep, things like that. So serotonin does have quite a few projections that impacts the cortex at many different levels. Okay, so we're going to end here, and I am going to get into the amino acid neurotransmitters at the next lecture, just to break these up just a little bit. And